Every great discovery begins with an idea, a gut feeling that says maybe, just maybe, we can do things differently. It starts with an infectious passion that inspires future generations and lives on through the never-ending search for answers. Tonight, the Academy of Medicine, Engineering, and Science of Texas honors these 4 innovative researchers. These are the recipients of the 2015 Edith and Peter O'Donnell Awards. Thomas Trey Westbrook, Medicine. Worldwide, over 1 million women will develop invasive breast cancer every year. Last year, nearly 400,000 died from the disease.
You have splicing data. You have the biopsy data.
At Baylor College of Medicine in Houston, Dr. Trey Westbrook and his team are using cutting-edge technology to find genes that play a pivotal role in cancer. He helped develop a genome-wide screening approach that identifies sensitivities in cancer cells. Through this technology, called RNAi screening, Trey has discovered that cancer cells have unique vulnerabilities. Exploiting this Achilles heel could lead to powerful treatments.
What our laboratory has been focused on is trying to understand how a given cancer gene unveils a certain weakness for that cancer. And we've been uncovering those systematically with the technologies we've developed and then leveraging those into new therapeutic opportunities.
Trey is particularly focused on triple-negative breast cancer, any breast cancer that does not express the ER, PR, or HER2 genes. About 20% of breast cancers are triple-negative, and the outcomes are poor.
My lab's been focused really for the past 7 or 8 years now on trying to both understand what are the genes that are the culprits, the drivers of that disease, but also to unveil the weaknesses, the vulnerabilities of those cancers.
Triple-negative breast cancers are the most difficult to treat. Frankly, that's the area where we have the greatest need for understanding basic mechanisms as well as key targets.
For Trey, the fight against cancer is personal. During a span of 18 months, while working on his postdoctoral fellowship at Harvard, his brother, mother, and father were each diagnosed with cancer. Trey's dad died less than 2 years later.
I wish I had more time with my dad. I wish I had received more of his mentorship as I got older in life, but I think it set me on a path that I've been very passionate about, and maybe hopefully early enough to make a difference.
When it touches you personally, it moves into really a mission that passes beyond just the professional. And I think that very much adds to the passion with which he pursues his studies.
There are encouraging signs of progress. 3 of Trey's discoveries have led to therapies in development or in clinical trials. Also, breast cancer may not be the only cancer impacted by Trey's research. Many other cancers, including lung and prostate, could benefit from targeting cancer weak points.
My mother is a great reminder that the clock's still ticking for other people. It gives us a lot of hope, but certainly the victory is not ours yet, so we're still working on it.
Haiyan Wang, Engineering. The computers that we can hold in the palm of our hands today are a testament of the evolution of materials and technology. And Dr. Haiyan Wang is on the forefront of that evolution. In an effort to push those limits, she's applying her expertise in materials science in new and exciting ways.
From Stone Age to Bronze to Iron Age, everything was driven by material discovery. We come into the new century, and it will be driven by new material discovery as well.
Haiyan and her team at Texas A&M University are using nanotechnology to create more efficient materials and working to prove these materials could one day be used to make smaller, more effective fuel cells.
A fuel cell, for the sake of simplicity, is like an active battery, but it generates electricity rather than simply stores it.
Our goal is to make the material so that the efficiency can be much higher and the operation temperature much lower.
see fuel cell electric vehicles that you don't have to plug into the wall. I really literally believe that her fuel cell technology will transform transportation.
Where much of conventional material science research has focused on developing the perfect material to gain the optimum results, Haiyan has taken a different approach. Instead, she and her team embrace the natural imperfections of a material and use nanotechnology to enhance the material's performance.
If you can incorporate some smart and unique nanostructured defects, put them into the right spots, you can make a perfect material even better, actually.
This revolutionary innovation has broad potential in electronic materials and devices, such as the development of more efficient superconductors that could transform the capabilities for practically anything that uses or transmits electricity.
So high temperature superconductor has many potential applications because the superconductor means carry electricity without loss. It means the materials will be able to carry the electricity with zero conductance or zero resistivity.
Haiyan's work is an inspiration to her colleagues and peers in the field. But for her, inspiring her students each and every day is her #1 priority.
I will continue on my material discovery efforts as well as my inspiration efforts. I will try to keep on inspiring new generations of material scientists to join the workforce, to continue on the material discovery, to make the world a better place to live.
Yu Meng Science. Uncovering knowledge is not just about asking the right questions, but being able to answer them. And for Dr. Yu Meng, finding those answers starts by going back to the basics. At her lab at UT Southwestern, Yu Meng has devoted much of her research to studying the fundamental processes of the human body, starting with the study of proteins and how they are transported in and out of the nucleus of the cell.
We're working with proteins that are very temperamental, very difficult to work with. We have to kind of coax them biochemically to crystallize so that we can make enough to study their functions.
When Yu Meng and her team focused on one specific protein transporter, it led to a breakthrough discovery.
This protein had this nuclear localization signal, and it just turned out that neurobiologists and neurologists had found that this protein was mutated in the familial form of ALS at that specific site.
For patients suffering from ALS or Lou Gehrig's disease, this could mean new and improved treatments thanks to a better understanding of this debilitating illness.
If we hadn't done this work, the neuroscientists wouldn't have anything to connect to what the neurologists had found in terms of where the mutations are. So we provided the foundation for them to understand the mutations that they found in the patients.
After making this discovery based on the way a protein is imported into the nucleus, Yu Meng and her team decided to focus on an exporter protein that labs all over the world were already fiercely studying.
CREM1 is best characterized nuclear exporter, and amongst the cargoes of CREM1 are proteins that are called tumor suppressors.
In certain cancers, it's been found that the level of CREM1, this export protein, is very high. So inhibiting that process has become very important as a potential therapeutic target for slowing or inhibiting cancer growth.
Instead of focusing her energy on the medicinal implications of CREM1, Yu Meng and her team decided to go back to the basics and study the 3D structure of CREM1 to determine its basic functions. The discoveries led to the creation of a new and improved tumor-suppressing drug by Yu Meng's pharmaceutical collaborators at Carifarm.
What her work has done is to enable these pharmaceutical companies, and one in particular, to develop a drug that has less side effects, be well tolerated by patients, and have therapeutic benefit.
Yu Meng's work has shone a light on the importance of studying the fundamentals and has opened the door to dozens of other discoveries that could be made from focusing on the way proteins are transported in cells throughout the body.
We have only studied one importer and one exporter. There are 20 of them in the family. They've got to be important, otherwise they wouldn't be around and they wouldn't have stuck around for that many years. If I'm going to solve a problem, pick a problem, do something in my lab, it's got to be important, it's got to make a difference.
Charles Collins, Technology Innovation. For centuries, doctors and scientists have been tasked with trying to diagnose illnesses. But finding out the root of a sickness can take days and even weeks. That means patient costs are higher and outcomes often aren't positive.
So what kind of sample are you running?
At Luminex Corporation in Austin, Dr. Charles Collins has invented technology that allows physicians to diagnose patients quicker and more accurately. Charles's invention, MagPix, allows doctors and scientists to run up to 4,800 individual tests in less than an hour.
Our multiplexing technology brings together our beads and the ability to do lots of different targets all in the same assay. You often don't know why somebody is sick, and if you guess one at a time, it takes you forever to walk through all the different things that could be making them sick. So our technology enables us to bring all those answers in a single test quickly.
MagPix received FDA clearance and was launched commercially in 2010. A year later, it received the prestigious Medical Design Excellence Award. MagPix is now in diagnostic and research labs throughout the world.
You'll hear examples of people who are using it for malaria testing, in Africa. They've actually taken the instruments out of main labs and been able to move it out more because of the robustness of the design.
Ruggedness and reliability is important to the researcher or medical scientist in downtown Manhattan, and is just as important to the researcher in New Guinea or Sub-Saharan Africa.
Charles's invention is also being used to test for anthrax and other airborne threats. Charles's research on digital microfluidics has also resulted in a contract with the Defense Threat Reduction Agency. He is currently leading development of a handheld, field-deployable diagnostic tool that can detect bioterrorism agents.
Then you may be testing a soldier where you want to know, maybe as a gastrointestinal, like what's causing the soldier to be sick, and that's the molecular panel. So you do this molecular test saying, well, what are all these markers that are making him sick? You may find C. diff, and you say, okay, well, that's probably what's causing that diarrhea. And so if you combine those two pieces of information together, then they can give a lot better profile of what's causing that soldier to have problems and how is their body responding to it.
Charles's contributions have advanced clinical diagnostics, healthcare, and scientific research. At Luminex, his MagPix technology has led to one spin-off product with another one in the works. Charles is just shy of 40, so expectations are high for even more discovery and invention.
The single most important thing to Chuck is to set ego aside and to make sure that we advance medical science, we make the world a better place. He saves lives for a living, and he takes that very seriously.
We spend years developing the product, but it's not till it gets out there into the market and people are doing these things with it that you really see, like, oh yeah, that's what I was doing this for, was to get something out there that gave them the capability to save these people's lives.
Congratulations to the 2015 recipients of the Edith and Peter O'Donnell Awards.